So isotypes are the antigenic determinants of the constant regions. They can be defined by antibody classes and subclasses. Isotypes are common to all the individuals of a species. For example, in case of humans, there are 5 heavy chain isotypic determinants, on the basis of which we call the antibodies IgM, IgD, IgG, IgA and IgE. And there are 2 light chain isotypic determinants which are kappa and lambda chains. What is the origin of isotypic determinants? Isotypes arise from the differences in constant regions of heavy chain and light chain. The changes in the genes or genetic arrangements in the constant regions, on the basis of which the functions of these antibodies can be modified. For example, in primary or first time infection, the first antibody will be produced, which is IgM. But if the infection persists or the body is infected with the same antigen more than once, there is class switching, and the class switching takes place into IgG. So IgM and IgG differ in the heavy chain. Both are directed against the same antigen but can perform different functions. For example, IgM is responsible for primary immune response. It is responsible for complement activation. But in case of IgG, there can be opsonization, neutralization, there can be placental transfer in case of pregnancy, and if there is class switching to IgA, if the site of infection is, for example, GIT, there is also presence of mucosal immunity. If there is allergic reaction, there will be class switching to IgE, and it provides function in various anti-inflammatory, anti-allergic, anti-parasitic interactions. Example, we can take the class switching of IgM into IgG, and this was about the isotypes, and isotypes are due to constant regions of heavy and light chain. Now let's talk about allotype. Now these allotypes can be defined as inherited antigenic variations in the constant regions. All the members of a species will have these 5 isotypes, but if we see closely the heavy chains of one of these 5 isotypes, we may find some kind of variations. We may find some specific sequences in some individuals and some specific sequences in another individuals. These differences among individuals of a same species will give rise to allotypes. Isotypes gave us the antibody classification. It distinguishes between various classes of antibodies, and allotypes distinguish between the genetical variants of one of these classes, of one of these isotypes within a particular species. If we talk about the structural basis, these allotypes are the allelic differences in immunoglobulin genes, and due to these allelic differences, when these proteins are translated, there are differences of about 1 to 4 amino acid substitutions, and these changes mainly occur in the heavy chains or light chains, but only on the constant regions. If we talk about different types of examples, we have GM allotypes on the gamma heavy chain. So we have different GM allotypes like GM1 sequence, GM3 sequence, GM17 sequence. All these 3 sequences are the allelic variants of IgG heavy chains. Similarly, on the light chains, we have KM allotypes, in which K stands for kappa marker. So we have KM1 sequences, we have KM3 sequences, which are occurring in the kappa light chains. Now suppose there are 2 persons and we are taking the same antibody, which is antibody gamma IgG. Now the person A and person B can have the antibody directed against the same pathogen, but it may have some variations in their allelic sequences. For example, a person A is having GM1 allotype while person B is having GM3 allotype, whereas there may be some person C which will be having this GM17 allotype. Suppose these IgGs are directed against the same pathogen, but what happens if we isolate this IgG from person A and we inject this IgG into person B? Will there be any immune response? Yes, there will be specific immune response because there are slight variations in the allelic sequence, and these variations will be recognized by the immune system of this person B. And due to these differences, these markers are used in various kinds of studies, like studies in population genetics, in anthropology, in forensic medicine, in various kinds of transplantation immunology. And if we talk about some clinical relations, suppose a fetus is being developed inside a mother's womb, and fetus inherits a different type of allotypic sequence from the father. Now, at the 7th month, the mother's immune system may produce antibodies, or we can say anti-allotypic antibodies. In such cases, intervention may be required. Now, let's talk about idiotype. Idiotype can be defined by the antigenic determinants in variable regions, and these variable regions consists of both heavy chain as well as light chain variable sequences we call CDRs, complementarity determining regions, and we have three such CDRs: CDR1, CDR2 and CDR3, and these three CDRs will make a specific idiotype for each antibody, and each antibody may have a different CDRs. That is why each antibody can have different idiotype. As we saw previously, the isotypes were common to a common species, allotypes, but the differences within the one species. And in case of idiotypes, it represents the individuality of a particular antibody clone. Now, what are the differences between idiotope and idiotype? If we talk about idiotope, it is the single antigenic determinant, and if we talk about idiotype, it is collection of all the idiotypes present on an antibody. And how does this idiotype arise in different kinds of antibodies? The answer lies in three different phenomena. First one is VDJ recombination, second one is junctional diversity, and last one is somatic hypermutation. We will study these processes in case of variation in antibodies, which is a separate topic altogether. Now, if we talk about examples, the two antibodies which are having the same isotype, for example, we take IgG, which is having the same isotype, same class, both are IgG, and suppose both are having the GM3 sequence, both are having the same allotype as well, but these antibodies may bind with different antigens. They may bind to different epitopes, which means they are having different paratopes, they are having different CDR sequences, and this variability arises due to these idiotypes, because both the antibodies, despite having the same isotype, despite having the same allotype, both the antibodies are having different idiotype. Different idiotypes acts as antigen inducers. If we isolate some kind of antibody from person A and we introduce that antibody into person B, there will be immune response against that particular antibody, and there will be formation of anti-idiotype antibody, we can say. If we talk about biological significance, it determines the epitope specificity and it provides the true basis of antibody individuality. If we talk about clinical importance, we have various B-cell lymphomas. Each malignant B-cell clone will express unique or specific idiotype, which can be recognized as a tumor-specific marker, and there can be developed monoclonal antibody therapy approaches. Idiotypes can be used to monitor responses and to design anti-idiotype specific vaccines or therapies. Now, what are the relationships, or how can we summarize this concept of isotype, allotype and idiotype? For isotype, it tells us the class or subclass of this antibody, whether it is IgM, IgG, IgA, IgD. Now, allotype, for example, we take an antibody IgG1. Now, this allotype will tell us about the specific variation within the species. For example, it is of GM17 allotype. So, it determines the genetically inherited variant of a specific isotype. Talking about idiotype, it will determine the antigen binding site. It will determine the paratope, the CDRs, and it will be providing the unique specific identity to that antibody. If we talk about the differences, or we summarize the whole concept, the basis of variation. In isotype, the basis of variation is the differentials in heavy and light chains, which gives us classes and subclasses of the antibodies. In case of allotype, there are the genetically inherited allelic variations within these classes. In case of idiotype, these are the unique sequences of the paratope, unique CDRs, which are determining the uniqueness of a particular antibody or a particular B-cell clone. Isotype is found in all the members of a species. For example, humans are having 5 different classes of antibodies. The allotype is found in some individuals of a species, because some individuals may show specific markers like GM17, and idiotypes are found in specific B-cell clones, because it gives a unique identity to each antibody. Let's talk about genetic basis. So, isotypes arise from different arrangements of immunoglobulin genes. That's how class switching takes place. Talking about allotype, these are the different allelic variations which will give rise to 1 to 4 different amino acid sequence. And in case of idiotype, we talked about 3 bases, which are VDJ recombinations, somatic hypermutations, and junctional diversity. If we talk about locations, the isotype is restricted to constant regions of heavy and light chains, and allotype are some specific variation between these constant regions of heavy and light chains, whereas idiotypes are present in variable regions of both heavy and light chains. And if we talk about specific sequences, we call those sequences CDRs, complementarity determining regions. Talking about examples, in isotypes we have different classes of antibodies. For example, in humans we have, if we talk about heavy chain, we have IgG, IgA, IgM, IgD, IgE. If we talk about light chains, we have kappa chain, we have lambda chain. For allotype, we have different allelic variations within the heavy chain. For example, we have different GM sequences. For light chain, we have different KM sequences for kappa chains. And if we talk about idiotype, examples are the specific B cell clones, specific antigen binding site of a specific antibody. Talking about biological significance, the isotype determines the antibody effector function. For example, the IgM was responsible in the primary immune response. IgG is responsible in secondary immune response. It provides other functionality like opsonization, neutralization. The IgE is responsible in the allergic immune responses. IgA is functioning in the mucosal immunity. So it determines the effector function. But kind of effector function will that antibody be performing against the same antigen? Whereas in case of allotype, it gives the genetic polymorphism. In case of idiotype, it determines the antigen specificity.